Welcome to our Bioprocess Engineering online tutorial on Bioprocess Environmental Lifecycle Assessment. Through the Bioprocess Engineering online tutorial series, you gain expertise in the principles of bioprocess engineering and their applications to enzymatic bioconversions, microbial fermentations, and cell cultures bioprocesses. For more details, please visit us at bioprocessengineeringonline.com. In this introductory tutorial on the environmental assessment of bioprocesses, we'll address three questions. What is life cycle assessment? How to assess the environmental impacts of bioprocesses? What are examples of bioprocesses impacts indicators? Life cycle assessment, or LCA, is a methodology for evaluating the potential impacts of a product on the environment throughout its life cycle. Life cycle assessment assesses the environmental effects of material and energy resources consumption and of emissions into the air, water, and soil. The primary focus of life cycle assessment is to evaluate the environmental impacts of a product across all its stages, commonly referred to as cradle to grave. These stages encompass the extraction, production, and transportation of raw materials utilized throughout the cycle, as well as the production and transportation of energy vector like electricity, fuel, and gas. Additionally, they encompass the manufacturing, packaging, distribution, and utilization of the product. Finally, consideration is given to the end-of-life treatment of the product. Upon reaching the end of its life, certain components of the product may be recyclable and repurposed as raw materials for manufacturing new products. In such instances, the sequence of stages in the product's lifespan forms a cycle, hence the term life cycle. Another essential aspect of life cycle assessment involves examining various environmental impacts. These include impacts linked to resource consumption, such as primary energy consumption, depletion of abiotic resources, and water consumption. They also encompasses impacts associated with emissions into air, water, and soil, including global warming potential due to greenhouse gases, formation of photochemical oxidants, and water eutrophication. Additionally, LCA may also address broader toxicity concerns, such as the potential for human toxicity. The LCA methodology, as recommended by standards, consists of four phases. The definition of the goal and scope of the study. The life cycle inventory, LCI. In this stage, data are collected on the inputs, raw materials, energy, and outputs, emissions, wastes, associated with each stage of the product's life cycle. Life cycle impact assessment, LCIA. Here, the environmental impacts indicators of the life cycle are evaluated and characterized. Finally, the results interpretation. The final stage involves interpreting the results of the assessment, drawing conclusions, and making recommendations for improvement. During the life cycle impact assessment stage, every environmental impact under consideration undergoes evaluation using specific impact indicators. Impact indicators serve as measurable variables that gauge the contributions of life cycle material and energy inputs and outputs to the impacts. Each impact indicator, denoted as IIMP, is derived by aggregating the impact contributions of all inputs and outputs according to the formula. IIMP equals sum of MI multiplied by FI, where MI represents the quantities of inputs and outputs from the material and energy inventories, and FI denotes the corresponding impact factors per unit quantity of input or output. The life cycle assessment of a product may focus solely on its initial bioprocess production phase, which involves the generation of material and energy resources. This bioprocess LCA provides an environmental footprint of the process. It proves invaluable in comparing various production methods for obtaining a product, such as biochemical versus chemical processes. By evaluating different approaches, it helps in identifying the most sustainable and environmentally friendly options. Moreover, LCA serves as powerful guide for environmentally optimizing bioprocesses, enabling the identification of areas where improvements can be made to minimize environmental impacts. For enzymatic, microbial, or other cellular bioprocesses, there are typically three major environmental impacts to consider. Firstly, primary energy consumption, PEC in megajoules. Bioprocesses often exhibit high energy consumption, particularly for the operation of fermentation and thermal unit operations, such as sterilization, evaporation, and drying. Secondly, water consumption, WAT, in kilograms water. Bioprocesses can entail significant water usage for media and buffer preparations, as well as equipment cleaning. Thirdly, global warming potential, GWP, in kilograms of CO2. In bioprocesses, there are notable levels of CO2 emissions during the fermentation stage and from energy consumptions. As a first step of the bioprocess LCA, the material inventory aims to identify and evaluate the quantities of raw materials, chemicals, and water utilized during unit operations and for equipment cleaning. Additionally, it assesses the solid and liquid waste generated as well as air emissions. The subsequent energy inventory evaluates the energy consumption of bioprocess unit operations. This encompasses electricity usage, such as for tank stirring or fluid pumping, as well as the consumption of natural gas or fuel to supply the heat required for the operation of sterilizers, evaporators, or dryers. The quantities of material and energy inputs and outputs assessed in the inventories are subsequently employed to calculate the three impact indicators, IPEC, IWatt, and IGWP. These indicators aggregate the contributions of materials and energy to each environmental impact. Impact indicators are typically computed, first for a batch operation of the biotransformation. Secondly, they are calculated per kilogram of produced product by dividing by the quantity of product generated. The assessment of bioprocess impact indicators requires the knowledge of the impact factors, FI, of the material inputs. These impact factors represent the impact indicators per kilogram of material obtained from a life cycle assessment of the respective inputs. They encompass all impact contributions during extraction, production, and transport stages. Their values are typically available in environmental databases. For example, for the glucose sugar, the impact factors are as follows: 25 megajoules per kilogram for PEC, 6 kilograms water per kilogram for watt, and 1 kilogram carbon dioxide per kilogram for GWP. The assessment of bioprocess impact indicators also requires the knowledge of the impact factors, FI, of the used electricity, gas, or fuel energy sources. These impact factors represent the impact indicators per unit amount of energy vector obtained from a life cycle assessment. They encompass all impact contributions during the energy vector production, transport, and utilization stages. For example, for electricity, the average European impact factors are as follows: 7 megajoules per kilowatt hour for PEC, 4 kilograms water per kilowatt hour for watt, and 0.23 kilograms carbon dioxide per kilowatt hour for GWP. In addition to computing the impact indicators per kilogram of product, the bioprocess LCA assessment also provides a ranking of the indicator's contributions from material and energy inputs and outputs. In bioprocesses, the primary contributors to PEC, watt, and GWP indicators typically include raw materials used in biotransformation, water consumption, CO2 emissions from fermentations, as well as from electricity and gas consumption. The industrial process for producing ethanol from sugar beets includes the following operations: washing and shredding the sugar beets, extracting the sugar, fermenting the sugar with the yeast Saccharomyces cerevisiae to produce ethanol, distilling the fermentation mash to recover the ethanol, dehydrating the ethanol to obtain a fuel. The ethanol production process generates two byproducts that are treated for valorization. Firstly, the beet pulps obtained at the end of the extraction operation. They undergo two operations of mechanical pressing and drying. Secondly, the distillery residues known as vinasses. They are concentrated through evaporation. The ethanol bioprocess material and energy inventories are established on a production plant with a 10 cubic meter fermenter. A batch operation utilizes 10,000 kilograms beets, 4,500 kilograms water. It produces 700 kilograms ethanol, 750 kilograms CO2, 550 kilograms pulps, and 400 kilograms vinasses. For the equipment operation, the process consumes 650 kilowatt-hours electricity and 450 cubic meters natural gas. Per kilogram of produced ethanol, the calculated impacts indicators are as follows. 40 megajoules per kilogram for primary energy consumption. Most important contributions are from gas, electricity, and from sugar beets. 22 kilograms water per kilogram for water consumption. The main contributions are from sugar beets production, extraction water, and electricity. 3 kilograms CO2 per kilogram for global warming potential. Most important contributions are from gas, electricity, and sugar beets. Enzymes are currently produced by fermentation processes using microorganisms, for instance bacillus bacteria, that secrete the enzymes. The enzyme production process by microbial fermentation involves the upstream operations of media preparation, sterilization, and fermentation. For accrued enzyme production, typical downstream operations are the centrifugation of the cellular biomass, the concentration of the enzyme solution by ultrafiltration, and the final spray drying for the production of an enzyme powder. The enzyme bioprocess material and energy inventories are established on a production plant with a 10 cubic meter fermenter. Under current operational conditions, a batch operation utilizes 1,200 kilograms glucose, 250 kilograms salts and minerals, and 10,000 kilograms water. It produces 350 kilograms of enzyme, 220 kilograms of CO2, 1,000 kilograms of solid waste, and 5,500 kilograms of liquid effluents. For the equipment operation, the process consumes 1,200 kilowatt-hours electricity and 500 cubic meters gas. Per kilogram of produced enzyme powder, the impacts indicators are as follows. 155 megajoules per kilogram enzyme for the primary energy consumption. Most important contributions are from glucose, natural gas, and electricity. 80 kilograms water per kilogram enzyme for the water consumption. Main contributions from glucose, water for media, and electricity consumption. 9 kilograms carbon dioxide per kilogram enzyme for global warming potential. Most important contributions are from natural gas, glucose, and electricity. Accordingly, for the three impacts indicators, it is the production of the glucose used in the fermentation medium that brings a dominant contribution to the enzyme environmental footprint. In conclusion, in this introductory tutorial on bioprocess environmental evaluation, you learned the life cycle assessment methodology to evaluate the environmental impacts of bioprocesses from materials and energy inventories, the three main impacts indicators evaluated for bioprocesses, the primary energy consumption, the water consumption, and the global warming potential impacts. If interested, you can gain a deeper insight in the LCA methodology by enrolling to one of our online courses. Introduction to Bioprocess Life Cycle Assessment and Life Cycle Assessment of Bioprocesses and Bioproducts. For further information, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com. We hope you found value in this introductory tutorial on bioprocesses. Your feedback is important to us, so please share your comments with us. Explore our website for more information about our comprehensive online courses in bioprocess engineering, and be sure to subscribe to our Bioprocess Engineering online channel for upcoming tutorials. Thank you for watching.